we are going to about our lipids. Hold on, just give me 1 second. So when we're going to talk about our lipids, right? So an easy way or an easy term in which we can define lipids is that they are basically your fats. Now, when we talk about fats, so fats, you might have heard of them in as fats, or you might have heard of them as an oil, right? Oil and fats and lipids, pretty much the same thing. They're all pretty much the same thing. The only difference is that your fats are solids, right? So your animal fat, your beef fat, your goat fat, your animal fat is what we call fat. And it's generally solid when it comes to its state at room temperature. And oils are generally derived from plants. So the lipids that we get from plants are considered to be oils. And of course, they're going to be liquid at room temperature. So lipids are organic compounds, just like carbohydrates, they are organic compounds, and they contain a lot of carbon hydrogen bonds as well. So they're really rich in carbon and hydrogen atoms. They do have oxygen atoms as well, but not as much, or in fact, you can say almost the quantity is almost negligible. It's like quite small. So they do have oxygen, but it's quite small, the quantity of it, right? So most of the atoms consisting or that comprise a lipid are your carbon atoms and your hydrogen atoms. Now, when we talk about lipids, so lipids have a wide variety of utilization, right? Why are they such an important biological molecule? Why did I emphasize on them, or why did I say that we consider them to be a major component of our body? It's because they have a lot of different functions. One of the major functions of lipids, when we talk about it, is that you and I need lipids for the purposes of energy. We can actually break down lipids and use it to provide energy, just like carbohydrates. In fact, lipids provide us with a lot more energy compared to carbohydrates. The energy we get from lipids is a lot more compared to the energy we get from carbohydrates. And the major reason why we get so much energy from lipids is because lipids have a lot of carbon hydrogen bonds. Because the reason we even get energy is by breaking these bonds. And since carbon and hydrogen are since your lipids are so rich in carbon and they're so rich in hydrogen, right? So, because they have plenty of these bonds, so when we break them, of course, the energy released is a lot greater than carbohydrates. But even though lipids do provide more energy compared to carbohydrates, they are not the primary source of energy. The primary source is carbohydrates. That's like my first. First, I will break carbohydrates, and in case where I have broken down carbohydrates or there are not enough carbohydrates in the body, then lipids would be the second that okay, now we will break lipids for the purposes of energy. Now, other than that, a really important function of lipids when we're talking about it is they have a role in insulation, right? So lipids, it's not only in just in human body. When we talk about biology, we often think of human body. But insulation is extremely important, not just in humans, but in animals, right? A lot of those animals that live in really cold environment, a lot of those animals that live in really cold environment, they are actually consist of layers and layers and layers of fat under their skin, right? It's often in most of the animals that insulation or that layers of fat is known as blubber. So they have these layers and layers and layers of fat under their skin, which helps in insulating, it helps in maintaining the body temperature, it helps in ensuring that the internal temperature of the body is maintained. Then another really important reason of lipids is that we need them for making a lot of different components of the body. For example, our cell membrane. And you and I have the topic of cell membrane in Unit 2. So we are going to study the structure in detail over there. But the cell membrane is composed of lipids as well, right? So they once again, you can see lipids have quite a major use. We need them to make the cell membrane. We need them to make the different vesicles that we can see within our cells. So lipids have a variety of different utilizes when it comes to inside our body. Now, let's talk about what is the structure of lipid. The structure of lipid is actually easy to understand, right? If you have a little bit of basic chemistry under you, this will become even easier to understand. When we are talking about organic chemistry, so if you've done that, this might make slightly more sense. But even if you haven't covered it before, it doesn't matter. Our topic remains the same. So our fats can be of different types. The most common type of fat that you will see in the human body, or the most common type of fat that you will see in your animals, are known as triglycerides. Triglycerides. This is just one type of lipid. Then we can have other type of lipid. We can have phospholipids, which we will also study when we do the structure of the cell membrane. Then we can have there are other types of lipids as well. So triglycerides though are like the most common type that you can have inside the animal body. Now, how do triglycerides form? For this, we need two molecules. The first molecule that I require is going to be an alcohol. It is an alcohol that has got three OH groups. So it has got three carbon atoms and it has three OH groups. Right? This alcohol, which has got three OH groups, so the biological name for it, the chemical name for it might be different, but the biological name for it is it's known as glycerol. Along with glycerol, we need another molecule. That molecule is, if you've studied, like I said, organic chemistry, you might know of carboxylic acids. If you haven't studied it, it doesn't matter, right? So we have a organic compound that has a very specific structure. This C double bond O OH, right? This group is known as a carboxylic acid group. That's the specific terminology for all compounds that have this particular structure inside it. Now, after that, we have a really long chain of carbon atoms, like a super duper long chain of carbon atoms. We don't generally draw that long chain of carbon atoms. We just draw, this is known as a skeletal formula. So we just draw, sort of we just draw a wriggly line to show that this is the long chain of carbon atoms that is present. This chain of carbon atoms can be 10 carbons long, 20 carbon long. It can be as long, as that particular structure requires it to be. This is a basically in chemistry, we would call it a carboxylic acid, but the biological term for it is, we call it a fatty acid chain. So we need one molecule of glycerol and I require three molecule of fatty acid. What is going to happen? These two are going to join together and they're going to make a bond. What is the bond even going to look like? The bond is going to look like this. So first of all, this is my molecule of glycerol. And then I have my carboxylic acid. I will require three carboxylic acid. Again, like I said, the carboxylic acid or the fatty acid can have as many number of carbons as possible. It doesn't affect, it's not fixed. And I will require three of these. Now, these two are going to join together. These are going to join together. So the reaction that's going to take place is going to be known as condensation reaction. Just like we studied with glycosidic linkage, the reaction that's going to take place is going to be known as a condensation reaction. So they're going to go ahead and a condensation reaction is going to take place. What is going to happen? The OH from the alcohol and the hydrogen from the fatty acid will combine with each other. And this will happen in all three of the groups like this, right? a bond will form between the alcohol and the fatty acid. A bond will form between the glycerol and the fatty acid. And this is what the bond is going to look like. So the OH from your alcohol is gone. Notice how I did not draw the OH from the alcohol. Why? Because the OH from the alcohol has reacted with the hydrogen from the fatty acid. We will be left with the oxygen of the fatty acid. And then we will draw the rest of the fatty acid. And this is what a triglyceride actually looks like. Right? And this particular, so in carboxylic acids, in carbohydrates, we had studied that the bond in carbohydrates is known as glycosidic bond. What is the bond known that exists between triglycerides? This bond that exists between triglycerides, this bond is known as an ester bond or ester linkage. In fact, if you have done organic chemistry, like I said, you might recognize that this is an ester. That's what it basically is. A triglyceride is basically an ester, which is a combination of your glycerol and a combination of your carboxylic acid to give you an ester. But again, you do not need background knowledge to understand this. This basically, it's one molecule of glycerol, three molecules of carboxylic acid, or three molecules of fatty acids. That's what you should say in the exam. One molecule of glycerol, three molecules of fatty acid. The OH from the alcohol reacts with the hydrogen of the fatty acid. An ester bond forms between the molecule of glycerol and your fatty acids. So you will get two products in the reaction. One product will be your triglyceride. The other product will be three molecules of water, each from each bond, right? Because you're making three bonds. Since you're making three bonds, so you're going to end up seeing that there are going to be three molecules of water that are going to be produced. So this is the basic structure that you're going to go ahead and you're going to see with your triglycerides. Making sense? Any confusion?
Oh no, everything's clear. Also, I took chemistry O level, so
Okay,
I'm
all right.
fine
So
with
then,
everything.
yeah, then I hope it's making like really good sense.
Yeah, yeah, it does.
Okay, now when we are talking about these fatty acid chains, right? So these fatty acid chains can be unsaturated or they can be saturated. Hold on, let me just type this out for you 1 second. So your fatty acid chain, right? Your fatty acid chain can be unsaturated and it can be uh, saturated as well. Now, when we're going to talk about it, what do we mean by saturated? Saturated means that it does not have any double bonds. Now, when we're going to talk about it, why are we even discussing this? Because the presence of double bond changes the properties of those triglycerides, right? If a double bond is present, that makes the triglyceride a bit more flexible. It makes it a bit more mobile, right? But if a fatty acid is going to be saturated, right? So it generally tends to have a bit more of a rigid structure. So it changes the properties associated with the fatty acid and as a result with the triglyceride. So it does not have any double bonds, right? Then on the other hand, or it can be unsaturated, in which case it's going to have double bonds present between the carbon atoms. If a molecule has only one double bond, so we call it mono unsaturated. Mono unsaturated. Mono unsaturated means that this particular fatty acid has only has only one double bond. That's it. And what if we have more than one double bond? If there are multiple double bonds present, in that case, we are going to have polyunsaturated, which suggests the fact that multiple double bonds are actually present. So our fatty acids can have different structure depending upon, what their required function is. So we can have saturated or we can have it unsaturated, and inside unsaturated then we have further options. It can either be mono unsaturated or it can be polyunsaturated. So this is the structure of what you need to know about your triglycerides. When we're talking about our triglycerides, so the reaction that joins the glycerol molecule and the carboxylic acid, I told you it's a condensation reaction. Another name that you can use for it is that we can also call it esterification, right? And then we can also go ahead and we can do the reverse of it. We can go ahead and we can do the opposite of it. What is the opposite of it? The opposite of it is that we can take our triglyceride and we can break it down to give us a glycerol and to give us a fatty acid. The reverse reaction is going to be known as hydrolysis. So the reverse reaction is known as hydrolysis. And the forward reaction, where you are joining the glycerol and fatty acid to give you carboxylic acid, sorry, to give you the ester, triglyceride, that is your condensation reaction. It's also known as esterification. So you have different type of lipids in the body. Like I told you, this is the most common one that you're going to be dealing with. There is another really common type that right now I'm not going to go into detail regarding it, but I'll give you basic structure of it because we have a whole chapter coming up in the next unit anyways. But I will give you basic information regarding it. So another in your syllabus, another really important lipid that you need to know about are phospholipids. What are phospholipids? So phospholipids are similar to triglycerides. In fact, they're quite similar to triglycerides, right? But there are small differences. Okay. One of the first, let's go over the similarities. The similarities is that it is two of the components are the same, meaning that it's going to have one component of it is going to be the glycerol, right? One component of it is going to be your glycerol, right? That portion is the same. In fact, you don't even need to draw the detailed structure of this. One component of it is exactly the same. We have glycerol. Now, just like we studied with the triglycerides, this molecule of glycerol is going to be attached to your fatty acids. The difference that comes is that the number of fatty acids are only two. In phospholipids, there are only two fatty acid chains. That's it. Not three. Triglycerides have three. That's why their name is triglycerides. Phospholipids only have two. Now, they have one additional component that we do not see with triglycerides, and that is a phosphate group. So they have a special group of phosphate, right? So we literally have a phosphate group which is attached to your glycerol molecule. So glycerol molecule has got three alcohol groups, like three OH groups. So two of those groups are going to be involved in making making an ester bond with the fatty acids. So two of the OH groups are going to be involved in making an ester bond with the fatty acids. The other OH group is going to be involved in making a bond with the phosphate group. Now, so this is a phospholipid. That's why the name is phospho, because it has got a phosphate element incorporated in the molecule. And why? So phospholipids are really, really important because they are responsible for our cell membrane. What is so special about them that allows them to function inside the cell membrane? Or what's important about them that makes them the main component of a cell membrane? The reason for that is, okay, now over here, I'm just going to give you a basic concept. We're not going to go over detail of it because that will take too much time. The reason for that is this phosphate portion is water loving. So this molecule is quite mixed. You know how, like, you have, for example, a molecule, it will either be soluble in water or it will not be or it will be insoluble in water, right? That's the kind of molecules we'll be dealing with, that it is all soluble in water or it is all insoluble in water. Phospholipids are somewhere in the middle. There are portions of the molecule that love to be in the water, that are highly soluble in the water. And then there are portions of the molecule that actually hate to be in the water. They're literally water hating. And what portion of the phospholipid is that? This, the fatty acid chains. They are water hating. They want to be as far away from the water as possible. So the phosphate portion loves being in the water. The terminology we use for it is, we call these sort of molecules or these kind of structures hydrophilic. Hydro means water, philic means loving. Water hating, so what's the term we use for water hating? Hydrophobic. Hydro again means water, phobic means hating. So the phosphate group is hydrophilic. In fact, we have a proper term for it when we talk about phospholipids. We call it the hydrophilic head. We actually have a name for it. We say this is a hydrophilic head. And the fatty acids, we actually call them hydrophobic tails. So this molecule has a really sort of a mixed situation going on. Some parts of it are soluble in water. Some parts of it are insoluble in water. But what's the purpose? Why have such a weirdly mixed structure? Why do we need to have, I mean, how is this molecule even going to exist, right? If parts of it want to be in the water and parts of it do not want to be in the water, how is this molecule even going to survive in our body? Now, okay, over here, the molecule arranges itself in a very specific way in our cell membrane. In our cell membrane, the molecule arranges itself in a very specific way. What happens that, let's say that this is my outside of the cell and this is the inside of the cell. Outside of the cell, we have fluid, the bodily fluids, your tissue fluids and all that. And inside of the cell, we have the cytoplasm. Now, what's going to happen? The water-loving phosphate group will say, I want to be facing the fluids. I want to be facing the cytoplasm. It will say, listen, I want to be facing the cytoplasm. I am water-loving. Now, the hydrophobic tails will say, listen, I want to get away from water as much as possible. So what are they going to do? They are going to try to point away from the water. They will try to, face away from the water. Now, generally, if we take a bunch of phospholipids and we put them in fluids, what will I see? If I take a bunch of phospholipids and I put them in a fluid, I will see that they will actually end up making a ball. And they will end up making the ball in such a way that the water-loving portion of the molecule will be facing the outside, the fluid portion, right? They will be facing the outside, the fluid portion. While the water-hating portion, all the water-hating tails will all face the inside of the ball, right? They were like, listen, we'll just huddle together. We will just come together and we'll sort of create this inner region where there'll be no water. And the hydrophilic heads that love to be in the water, they can face the water, they can be inside the water. The rest of us, we're going to come in the middle of the ball. We're going to all huddle close to each other so that we can create this sort of a region where there's no water present, right? So this is how phospholipids would naturally act. You take a bunch of phospholipids, you put them in water, this is what they would do. They would make literally a ball. And they would arrange themselves in such a way that all the hydrophobic tails will be away from the water towards the center of the ball, and the hydrophilic heads would be facing the fluid. In the cell membrane, though, we do not have a ball-like structure. In the cell membrane, we actually have something known as a bilayer. What do I mean by bilayer? It means that I will have two layers of the phospholipids. So this is one layer, and then I will have the other layer. And the two layers will arrange themselves in such a way that the hydrophilic heads are going to be facing the fluids, right? So the hydrophilic heads are going to be facing the outside of the cell, because outside the cell you have fluid. The hydrophilic heads will be facing the other layer. The hydrophilic heads of one layer will face the outside of the cell. The hydrophilic heads of the second layer will arrange themselves towards the cytoplasm. And what will happen? The hydrophobic water-hating tails of both the layers will arrange themselves towards each other. They will arrange themselves towards the center of the membrane. They'll be like, listen, we'll all just sort of huddle together, point towards each other, and come towards the center of the cell membrane so that we are away from both the cytoplasm as well as the fluid that's existing outside the cell. So this is how phospholipids make your cell membrane. And if you think about it, so what's going to happen? The tails are pointing towards each other. These tails are water hitting. So the center of the cell membrane actually becomes hydrophobic. It becomes water hitting. If you remember your IGCSE biology, you might have studied one of the major functions of a cell membrane is to what? Control who is coming into the cell and who is going out of the cell. This is how it does it. Anything that's water soluble, anything that is dissolves in water, your ion, sodium ion, potassium ion, chloride ion, glucose is water soluble. Any large molecules that love to be in the water, they cannot directly cross the cell membrane. Why? Because the center of the cell membrane is hydrophobic. Why? Because of this special arrangement of phospholipids. So Nothing can enter the cell or exit the cell on its own. It's not to say nothing. Oxygen, carbon dioxide, really, really small molecules, they can travel through the cell membrane, but large molecules and especially water soluble molecules, sodium, chlorine, chloride ions, potassium ions, glucose, these large water soluble molecules cannot directly either enter a cell or exit a cell. For that, you would require special proteins to help with the transport. We will study all those special transport proteins when we do the topic of cell membrane, right? So this is how the phospholipids are going to be arranging themselves. And this is why the cell membrane is able to regulate, and the cell membrane is able to make sure that, nothing enters or exits the cell unchecked. It will only enter and exit the cell by using those special transport proteins, and that's it. Right? We understand, making sense regarding this?
Yes, all is clear.
All right, great. So this is the topic of lipids. It's quite small. So we are done with the portion related to lipids. So now let's actually take a look at a few questions on our topic. Give me a second. Let me just share the screen with you. 1 minute. So look, we have this interesting question over here. The question says triglycerides found in living organisms have different melting points. Two factors that affect the length of a triglyceride are the length of the fatty acid chains, whether the fatty acid chains are saturated or unsaturated. A triglyceride is made from the components glycerol and fatty acids, which you and I have already studied. Stearic acid and arachidic acids are both fatty acids. Arachidic acid has two more carbon atoms than stearic acid. So arachidic acid is longer in terms of length, as compared to our stearic acid. The melting point of stearic acid is 69 and the melting point of arachidic acid is 76. Calculate the percentage increase in the melting point resulting from the presence of resulting from the presence of two more carbon atoms. Give your answer to 2 decimal places. Now, when you are dealing, you're going to see quite a lot of questions like these. Not just in your paper 3, also a lot in your paper 1 as well, where they will give you numerical data and they will expect you to be able to um, go ahead and actually calculate the percentage increase either from the numerical data provided to you or from the graph that has been provided to you. So let's go ahead and see how we can solve questions like this. Or if you have an idea of how you could solve this, you can let me know that I know how to solve this and I would like to try this on my own. So you could let me know then as well.
I think I still remember the equation. It
The
was
equations part, you want to give it a try?
yeah, it's so we do final, subtract from initial,
Yes,
and then
correct.
divided by the initial, and then we just times by 100.
Yes, correct. Okay,
So
good. So give that a try. Let me know what answer you're getting.
I got 10.14%.
Yes, that's absolutely correct. Good. Well done. Now, the next question says, devise a method to investigate how the length of fatty acids and the degree of saturation of fatty acids affect the melting point of your triglycerides. This is just a 3-mark question. In when you do paper 3 questions, might see similar questions as well, but they generally tend to carry a bit more marks. This is your paper 1 question, unit 1 question. So you don't have to give a very detailed answer over here. In paper 3, when it's going to be carrying more marks, they will expect more information from you. Like if you remember when we discussed about carbohydrates, the experimental portion of it. So I told you about independent variable, dependent variable, control variables, your methodology. So these things you are generally going to have to make sure that you add in your answer in paper 3 questions. In paper 1 questions, just giving a basic conceptual idea of how you're going to do the experiment is good enough. So would you like to go ahead and give this a try? Think of how, like I said, don't need any details from you. Just need a basic sort of an experiment that you can run.
I don't know. I'm not quite sure.
Yeah, all right. No problem. Absolutely no problem. So when we are going to, okay, so in questions like these, it is actually not as complicated as it seems. You need to figure out what is the effect of length of fatty acids and the amount of saturation, right? These two things. So when we are going to go ahead and talk about it, you need to figure out the role of it on melting point. First of all, let's talk about the melting point, right? When we talk about the melting point, so point number 1, you need to decide how you're going to run your experiment. So we will have to take our fats, we will have to provide some kind of heat, and we will have to note down the point at which they are going to change state, at which they're going to turn from solid to liquid. So that's the basic method I will have to follow. I will have to take my fats and I will have to um I will have to um I will have to melt them, provide heat energy. I will have to melt them to go ahead and I will have to note down the temperature at which the change of state takes place. Now, I will give you a basic answer for this as well. And just because we are practicing, I will give you a little bit more detailed answer in terms of what you can write down in unit 3. So we have got the basic methodology down. Let's mention our dependent variable. Our dependent variable is the melting point, right? And my basic method is I will take my fats, I will start putting them, start providing them heat with whatsoever method. You could use a water bath, you could put them directly on a Bunsen burner, whatever you prefer, and then you can note down the temperature at which the melting starts to occur. That's your melting point, right? That's our basic method. What is our independent method? A variable. You and I need to figure out the length of fatty acid and the saturation. These are the two variables that are in my control where I can go ahead and I can change them. Now, there's one problem. I can only test for one thing at one time. So what I can do is that I can select multiple fatty acids that have the same length, but they have different degree of saturation. So select fatty acids with the same length, but different degree of saturation. Heat up those fatty acids one by one and note down the melting point of each fatty acid. Then we'll repeat it, but with same saturation and different length. Then repeat with fatty acids of the same saturation and different length. Right? And again, note down the values of melting point. Repeat these experiments and calculate the mean. So you will repeat the experiments, calculate the average value, and you are done. Right? This is the kind of answer you can give in a paper 1 question. It's 3 marks, do not need to provide any detail. A basic method that I will keep the length same, but use different degree of saturations and note down the melting point for each one. Repeat it with same saturation but different lengths and calculate mean for both values. And once I have the data for both length and saturation, I can easily see what is the effect on the melting point, right? Now, when we are going to go ahead and we are going to talk about it, this is sort of a basic answer you can give in your paper 1. If you are dealing with a question that carries slightly more marks, so you can modify your answer to include, for example, control variables. Control variables are variables that you need to keep fixed, or you need to ensure that they are fixed for all the experiments that you're running. For example, the temperature of the water bath. For example, you're using a water bath to melt your fat. So the temperature of the water bath has to be maintained. When you're going to go ahead and talk about it, you need to maintain that. The sample, the amount, mass of fatty acid being taken. So each time make sure that you use the same mass of it, right? You have to make sure that you use the same mass of the fatty acid. You have to ensure that, it's adequately an insulated situation. All of them are, insulated in exactly the same way. You don't want one of them to be insulated more. You want all of them to be insulated in exactly the same way, right? You want to make sure you use the same thermometer, you make the same apparatus. So if you can add a few control variables in your answer as well. And then if you want, you can maybe also mention that you could draw a graph or to actually see the relationship between the dependent variable and the independent variable. Making sense? We understand how to answer these questions?
Yeah.
Okay, great. Now, again, similar question. Triglycerides found in living organisms have different melting points. Two factors that affect the melting point of a triglyceride are the length of the fatty acid chains and whether the fatty acid chains are saturated or unsaturated. A triglyceride is made from the components glycerol and fatty acids. How many of the following statements about triglycerides are correct? A glycerol molecule is joined to each fatty acid by an ester bond. What do you think? Is that correct or incorrect?
Correct.
Correct. An unsaturated fatty acid has at least one double bond between two carbon atoms. Correct or incorrect?
Correct.
Correct. All triglycerides have three fatty acids joined to glycerol.
Yeah.
That's also correct. So all three statements are correct. Our answer will be option D. The table shows the relative atomic mass of the elements present in triglycerides. How much lower is the molecular mass of this triglyceride molecule after it has been made from its components compared with the sum of its component molecules? What are the questions saying? If we take the molecular mass of the triglyceride and we take glycerol and carboxylic acid on its own, what is the difference in molecular mass? That's what they're saying. Our product is triglyceride, our reactants are alcohol and carboxylic acid. So if we take the molecular mass of the product and we take the molecular mass of the reactant, what's the difference in molecular mass? This question is much, much, much easier than it seems. And it's actually quite similar to a question that we did for the topic of carbohydrates as well. Would you like to give it a try?
Yes, just give me a few minutes.
Yeah, no need. Take your time.
Oh, okay. It's simple. I didn't recognize. It should be A, right? Because the water molecule, I think.
You're
Wrong?
you're on the right track, but
Okay.
let me give you a hint. If you go back to the reaction that we studied, how many molecule of water were we getting
Oh,
from one reaction? Yeah.
okay. So we times by 3.
Yes, you times by 3, and then your answer comes out to be?
54C.
So the answer is 54.
Okay.
Okay, question says glycerol molecules and fatty acid molecules are used in the synthesis of cell membrane, right? Just like we studied. The diagram shows a molecule of glycerol and a molecule of fatty acid. This graph was sketched. Hold on, let me actually go over this question. I feel like this might be a. Okay, no, we can actually go over this. This graph was sketched by a student to show how the membrane fluidity changes with temperature. Describe the relationship between membrane fluidity and temperature as shown by this graph. Now, graph questions are really, really, really common in your exam papers. Paper 1 especially, they're really common. And graph questions, so there are certain things that you need to remember while solving them. There are few points that if you remember, you can include them every single time, and that just makes it easier for you to write down the answer in the exams. Point number 1, always notice what the question is exactly saying. Does it say describe the relationship or does it say explain the relationship? Describe the relationship means that they do not expect you to use your theoretical information to answer the question. They expect you to only utilize the graph that's been given to you, meaning your information, your answer should all come from this graph. If they say explain, that means that not only do they want you to first describe the graph or tell what's happening in the graph, but then also give an explanation of what is happening by using your theoretical knowledge, right? This question says describe, which means we do not need any theoretical knowledge to solve it. We need to look at the graph. Now, from the graph, what you're going to do is, of course, you need to describe what you can actually see. This graph is relatively easier for us to deal with because they have not really provided us with any numerical data. But if you have been given a graph where there is numerical data, there's values in x-axis, there's value in y-axis, you have to give data from the graph. It's important. Include data from the graph. Point number one. Point number two. When you're point number two is that over here again, it's a simple graph. But later on, we're going to do graph where you're going to have to talk about, for example, we have something known as error bars, right? When we do those graphs, I will explain to you what they are. But you need to talk about those error bars as well. This question is simple. I don't have to worry about that. So you have to mention error bars. If given to you in the question, you have to talk about error bars. Then do take a look at the steepness of the curves or the steepness of the lines given to you because you can use them while giving your explanation. For example, we have to give the relationship between the fluidity of the membrane and the temperature. As the temperature increases, the fluidity of the membrane increases. That's a blanket statement. It's a general statement. It's not too descriptive. I'm not talking about exactly what's happening in the graph. I've sort of given an overview of what's happening, like one line overview of what is happening. So we generally, when we are giving our answers, we start with like a blanket statement that as the temperature increases, the fluidity of the membrane seems to be increasing. A blanket statement. Then I will start going more specific, right? As the when you're going to go ahead and talk about it, so you can say that as the temperature increases, the membrane fluidity shifts from a solid-like state to a liquid-like state. Now, between solid-like state and the liquid-like state, there is a sharp increase in fluidity because you will see that in over here in the solid-like state and liquid-like state, that's why I said we always pay attention to the steepness of the line. The lines are a bit flat. In the middle portion, the line's like really steep. It's quite close to being vertical. So what does that shows us? That shows us that for a small increase in temperature, the membrane fluidity has shot up. So I will incorporate this in my answer. I will say between the solid-like state and the liquid-like state, the membrane fluidity increases greatly for a small increase in temperature. I will mention that because like I said, the shape of the graph is important. So this is a 2-mark question. Those are the 2 marks. Point number 1, blanket statement. Overall, as the temperature increases, the fluidity of the membrane increases. Between the solid-like state and liquid-like state, there's a sharp and sudden increase or there's a great increase in the membrane fluidity, despite a small increase in temperature. And those are the 2 points you can give, and your graph is now completely described. Now, if this was explained, then you would have to add your theoretical knowledge, which you don't have at the moment because you have not done the topic of cell membrane yet, right? When we do the topic of cell membrane fluidity is something we are going to study. How is it actually maintained? How does this process happen? So when we do that, you'll understand the theory behind it. You'll be able to explain the relationship as well. But for now, when you're describing, you're going to incorporate these. So start with the blanket statement and then start going over if you have been given any coordinates, talk about coordinates. If you have been given any error bars, you mention error bars. If you have been, if they have talked to you about, your if they've talked to you, about if the shape of the graph is obviously in front of you. So if you can see any noticeable differences in how the shape of the graph is changing, you incorporate that in your answer as well. All right, making sense? All clear?
Yeah, it all makes sense.
Great. Okay, Asma, we'll go ahead and we'll stop it over here for today. And now, inshallah, in the next class,